Well, first, I'd like to say thank you to the session and workshop organizers for inviting me to give this talk. I'm happy to be here. I'm Sylvia Lee. I'm at the U.S. Environmental Protection Agency headquarters based in Washington, D.C. I am fortunate to work with some great colleagues there who helped develop this project. All of us pictured here are in a part of the EPA called the Center for Public Health and Environmental Assessment, and our group does research to support evidence-based decisions and actions to protect our environment, public health, and well-being. And I thought it was important to tell you about our group to provide some context for our approach for this particular project, where we're using information from the literature, data sets, and models, which are types of evidence that we can assess and synthesize to answer our research questions. And more broadly speaking, we're interested in kgml because we often work with both data-rich and data-poor sources of information, especially about ecological systems and field observational studies. And we'd like to know how we can put this information to work more efficiently. And it's important that we use information more efficiently so we can address widespread environmental problems like nutrient pollution in lakes. According to EPA's National Lakes Assessment, about 2 out of 5 lakes have excess total phosphorus concentrations. And this map here shows all the lakes that were sampled in the 2012 NLA, where the red points are lakes classified as most disturbed based on total phosphorus. And the NLA also found that these lakes in the red were more than twice as likely to have a degraded benthic macroinvertebrate community, which indicates that aquatic life is not well supported in these lakes. And a visible response that gets a lot of attention in response to excess nutrients in lakes is cyanobacteria blooms, which are increasing not just in the US, but around the world. This figure is from a study that used satellite imagery data from 71 lakes to determine bloom intensity based on the infrared signals, or to put it in really simplistic terms, how green the lake can get in a growing season. So this peak greenness has increased in many lakes, with the lakes indicated by the red triangles having the strongest increasing trend since the 1980s. And another study went back further in time using lake sediment records. This figure shows the concentration of cyanobacterial pigments in those sediments, which are stable for thousands of years once they are in the sediment and can be used as biomarkers of past algal biomass. The concentrations have increased rapidly over time, and this trend was best explained by nutrients and secondarily by temperature. There are some lakes where blooms continue to occur despite reductions in point and non-point source loading of nutrients. One example is Lake of the Woods. It's a large lake overlapping the border between northern Minnesota and Canada. I'm going to have several examples of lakes from Minnesota. And this lake used to receive phosphorus loading from paper mills and wastewater, but since the Clean Water Act, those sources have decreased, so the loading has decreased. Yet still, the lake turns green with algal blooms every summer. There's just so much phosphorus from past loading that's still in the lake and the lake sediments that the internal sources are feeding the blooms and remains a challenge for lake management. Better understanding of these internal loading and legacy phosphorus dynamics could help managers, especially if we could translate those phosphorus dynamics into estimates of recovery times. And what recovery time actually means will vary by lake and stakeholders involved, but having some estimate of when improvements to phosphorus concentrations could be detected given the internal phosphorus cycling, could help managers set reasonable goals and stakeholder expectation. An example of that is another lake in Minnesota called Bartlett Lake, where information about recovery time helped the local community make decisions about how to spend their resources. So in the figures here are diatoms from the sediment record of Bartlett Lake. And because diatoms preserve water in lake sediments, we can infer based on the diatom species what the total phosphorus concentrations were like before any lake monitoring data became available. And you can see, even without being an expert diatomist, that there has certainly been a change in the community composition from the 1880s and to modern times. So in this figure, the y-axis is the year, with the most recent year at the top, and going back further in time towards the bottom, similar to the age of the layers you'd encounter in a sediment core. And on the top is the x-axis with diatom inferred total phosphorus. The diatoms show that Bartlett Lake used to meet the state's standard in terms of total phosphorus concentration. And over time, there's been a gradual increase in abundance of diatoms, indicative of nutrient-enriched conditions. But point sources were removed in the 1970s, yet there has not really been much improvement in the diatoms because the diatoms are still responding to those phosphorus levels from internal sources. On the other hand, measured water quality, shown here in red, is slowly improving from over 120 micrograms per liter to currently around 40 micrograms per liter TP. And even though the recovery is pretty slow, phosphorus in the lake is continually being buried in the sediment layers. And based on the burial and the external load rate, that active pool of phosphorus will likely be depleted by around 2025, 2035, which will then be around 60 years of recovery time since removal of those point sources. And these estimates help the lake community decide that the best use of resources would actually be to have a bit more patience, as it seems that the lake is improving, although slowly. And to obtain this information, it was necessary to do a fairly extensive paleolimnology study and a phosphorus budget analysis for this single lake. However, that would not be feasible to do the same for a very large number of lakes. And that brings us to our research questions. The first question is a literature component. What are the important factors driving internal lake P cycling, and how do the relationships differ by lake ecosystem parameters? And what are the magnitude and consistency of these relationships? The second question is the modeling component. And we ask, if external phosphorus load into a lake is reduced by X%, how much time would it take for surface water TP concentrations to decrease by Y%? And these two components will inform each other, and together they'll be used to create an evidence base and model underlying some form of national map of recovery times for lakes across the US. So diving a little deeper into the literature component, this part is led by Kate Schofield. She developed this conceptual diagram that lays out the relationships among factors driving lake phosphorus cycling. And we found the paper by Jansen et al. Was helpful for developing the general framework to capture the relationships that are important for lake modeling. The framework consists of the four major components you see here in the diagram: watershed, climate, hydrologic, and the lake ecosystem component. So from that more simple diagram, here's the full version that expands the components into individual parameters that we will search for in the literature. And just as an example, I highlighted a pathway that could be the focus of study in a paper where nutrients from the watershed combined with wind and lake water depth are factors that influence resuspension rate and phosphorus concentrations in the water column. Along with the conceptual diagram, we conducted a literature search using citation mapping and a more traditional Web of Science search. Citation mapping is a forwards and backwards search of the references in a selection of seed references, which are papers we thought would refer to or would be referenced by other papers that captured the relationships we included in our conceptual diagram. And so from the thousands of papers we found in our search, we screened the title and abstracts and narrowed down the pool of papers to a much more manageable number of 240 references. And I wanted to show you something here that I thought could be of interest to the workshop participants. We recently started using machine learning in our literature-based projects to more quickly find relevant papers with text mining software like a Swift Active Screener. And there is also an option to use seed references, which provides the knowledge base to give the machine a head start in learning which papers are relevant, much like what happens sort of conceptually in kgml. So after the screening process, we use a spreadsheet like this one to extract information from the literature. And if that looks a little overwhelming and a lot of work, that's because it is. It's a challenging task to distill the pieces of information we care about from each paper and documenting them in a form that makes it possible for a meta-analysis or some other synthesis in a transparent way, and can eventually be made accessible to many people rather than sitting in the computers of our team members. Most of the columns here in the spreadsheet relate back to the shapes in our conceptual diagram. We also capture information about the citation, the study design, data analysis and results, and conceptual information that can help us with interpreting the data. And it was also important to capture specific water body characteristics so that the information from the literature can inform our modeling effort. So the modeling component is led by Jim Carlton. These are the mechanistic model equations that Jim has adopted from a model described by Chopra and Kennelly that represents the lake system as having two compartments, the water column and the benthic layer. And earlier, we already saw a three-compartment model of Lake Mendota from Paul's introduction. I'll note here that because we'd like the model to be applicable to many lakes, and because many lakes have little or no data, our strategy right now is to use a relatively simple and generalizable model of lake phosphorus cycling, even though we do recognize that lakes are usually more complex than two compartments. So this relatively simple model starts with a steady case, steady state case, and then simulates a dynamic case, which I'll illustrate with an example. So this example is based on Shigawa Lake, yet another lake from Minnesota. And here we assume the influent and water concentrations are constant and in equilibrium with each other. We also have some basic data like water residence time, the water TP concentration, mean water depth, and the depth of the benthic layer. And we also have these coefficients, rate coefficients for the model, including settling rate, recycling rate, and the burial rate. Then we can simulate what happens to the water column total phosphorus after this equilibrium is perturbed by a sudden decrease in influent phosphorus. So let's say that a sudden decrease in phosphorus loading is by 80%. And we can also set a hypothetical target concentration that represents potential recovery of the lake as 10 micrograms per liter. The resulting output is the graph on the right here with TP on the y-axis and number of days along the x-axis. And for this example, it takes over 50,000 days or 137 years for this hypothetical recovery. So we can use the same calculations for many lakes, such as those from the Lagos NE dataset of lakes in the Northeast and Midwest US. Here we simulated the same 80% drop in influent TP for these lakes. And the colored circles are lakes that will reach our hypothetical target concentration from ranging from less than 1 year to over 150 years. And from this output, it's nice to see that many lakes could recover in less than about 30 years. And we also have lakes in the Northeast where they are indicated by green triangles. And these lakes have already reached that target concentration at time 0. And finally, you have some black diamond points here indicating lakes in more agricultural areas that mathematically will not reach the target concentration even with infinite time. So in this example, we simulated less of a drop, only 20% in influent TP, which of course results in more of these black diamonds. But perhaps it gives us a little bit of hope about these lakes in blue or purple, where even a 20% decrease in loading could improve the water quality in less than around 20 years. So in summary, nutrient pollution impairs many lakes, and internal loading remains a challenge, even when there are external load reduction efforts. We're using information from the literature and building a recovery time model to provide evidence for lake managers who may be able to use this to develop long-term plans. And we also want to make this data available widely. We're interested in learning about transfer learning, putting information to work in novel ways, and melding knowledge with machine learning, things that I'm not that familiar with. And just that this project is in its beginning phases. And thank you to Paul and Chris for their collaboration and providing insights. We welcome any feedback about this project and ideas about how KGML could enhance the research. So thank you, and I'll take questions if there's time.
Thanks, Sylvia. A couple of questions, probably just time for one. So I'll go with the one that was asked first. So this says, often our models approximate behavior for a population of lakes, but citizens might care more about, their specific lake. Is there a way to resolve these two views?
That's a big question. Yeah. We're trying to, with this project, get more at that population scale, but we do recognize that there are lots of variability among different lakes, and it is really important to consider where these more general models don't work as well for those specific lakes, and see how we can reconcile those. So I really don't have an answer for that question.